Thank you, everyone, for sticking through this day. It's been really wonderful to hear from so many people on many topics that are very much in conversation with what I'm working on. Hello. My name is Riley Taitingfong. I'm a PhD candidate in the Department of Communication. I am so excited to talk to you a bit about my thesis work. And I'll just position myself a little bit in how I approach this work. I approach it as somebody with both Indigenous and settler ancestry. I'm Chamorro on my father's side, and European on my mother's side. I grew up in the Chamorro diaspora in the Pacific Northwest, and also was born in Spain, which might sound weird because they're like one of our colonizers, but we're here in the US. And also there's a US naval base there, which is why I was born there. So what you just heard in the last presentation about our people being so overrepresented in the military, this probably makes more sense. So the title of my presentation today is Editing Āina, Appropriations of Pacific Islands as Laboratories. And as you'll hear, my work looks at this emerging technology in genetic engineering known as gene drive. And I'm looking at broad questions about how the science of gene drive really participates in this figuring of islands as test sites, as a site of experimentation. And in following this technology in the Pacific, I've done much of my ethnographic work in Hawaiʻi. So I'll frame some of my key questions for you, just give you a sense. I won't get to everything today that you'll see on here, but just to give you a sense of things I'm grappling with in this work. One question is, how do new methods in the genetic engineering of non-human species, while kind of promising these revolutionary solutions in issues of conservation, how do they encode particular ideas about indigeneity and immigration? Another question I'm grappling with is, what are the everyday practices and imaginaries that scientists engage to organize gene drive research, including this imaginary of islands as isolated and contained? A third question, how do extant and emerging processes of regulation and governance shape gene drive research? So with this, I'm looking a lot at public engagement, how community engagement is being organized, and how that's shaping decisions about whether to release organisms with this genetic modification that I'll talk about, especially on islands in the Pacific. And lastly, how do these processes that I'm looking at reproduce settler colonial power relations, and how are they entangled in militarism? So I'm going to give you a background on this technology called gene drive. Has anyone heard of gene drive that didn't hear from me? So I think this will be new for a lot of folks. So I'll give you an intro to that and talk about this notion of islands as testing grounds that very much circulates within these discourses about gene drive. And then I'll talk a little bit about some of the findings from my work about gene drive in Hawaiʻi. Okay. So gene drive is a technique of genetic modification, and essentially it works by driving or pushing a specific genetic modification through an entire species of organism. And using CRISPR-Cas9 genome editing, scientists are able to use gene drive in ways that are really unprecedented, in that they're able to circumvent typical Mendelian inheritance. And we'll break down what this means a little bit. So let's imagine you have a genetically modified mosquito represented in the neon, and it mates with a wild type represented in the gray. And think back to Mendel, the peas, and the Punnett squares, and you'll remember that about 50% of the offspring are going to have that genetic modification. But under gene drive inheritance, this is going to be propagated through nearly 100% of offspring, right? So offspring are going to inherit the modification and the drive to keep propagating that modification into future generations. So how are scientists hoping to apply this breakthrough in gene editing? Lots of ways. There are applications across public health, conservation, and agriculture. For instance, gene drives being developed in mosquitoes to deal with the diseases they vector to humans, so Zika and malaria, and also the diseases mosquitoes vector to animals like avian malaria, which is the focus of gene drive for conservation in Hawaiʻi, for the conservation of endemic birds like the honey creeper. And there's also being work done to use gene drive to immunize mice to Lyme disease and eliminate entire populations of invasive rodents from islands. So I thought I'd give some context about the top funders of this research. We have the Bill and Melinda Gates Foundation, and these numbers are probably a lot higher now because these were as of 2017. We have the Bill and Melinda Gates Foundation at over $75 million for research into using gene drive as a malaria intervention. Also, the Tata Trust of Mumbai, $70 million for similar research into malaria, using gene drive for malaria. And a lot of this work happens in partnership with UC San Diego. So a lot of gene drive research happens here on this campus. And last but definitely not least, the US-based military agency DARPA has invested, I'm sure far more than this by now because they're in their second phase of the project, but $65 million toward research on gene drive. And we'll get into what's up with this a little more on the next slide when we talk about ethical issues. So as maybe you're already realizing, a technology that could potentially edit an entire species for generations and generations has a lot of ethical issues associated with it. So among these include public acceptance. This is a really big one. As I said, gene drive hasn't been released in the field yet. It's still in the lab. So there's a lot of conversation around the importance of gaining public acceptance or some kind of collective consent before it will ever come to the field. There's also a lot of concerns around regulation, which bring to the fore issues of international regulation. You know, what if you have gene drive mosquitoes in two different countries, you know, might have different policies about regulating these organisms, but the mosquito is not going to stop at our sort of arbitrary political boundaries, right? So a lot of issues around regulation. A big focus of my work is impacts on ecosystems. So we really don't know, and we can't predict with much certainty what's going to happen to ecosystems when gene drive is released into the environment, right? So some gene drives, like I said, are meant to propagate endlessly. And also some gene drives function by engineering population crash or even extinction. So there's a lot of concerns about, you know, unintentional release. A single mosquito could impact ecosystems, not just locally, but globally, right? And there's also concern about bioterrorism. This is a particularly scary one. So when we think about the fact that gene drive is potentially irreversible, we don't know, you know, the downstream ecological effects, and that it could spread on a global level. It's super concerning when we compound these issues with the fact that, you know, in theory, while gene drive could be used to prevent disease, it could be also used to spread disease, right? So a lot of DARPA's work focuses on, you know, anticipating these issues, trying to come up with countermeasures for gene drive, which for me sort of conjures up disastrous images of trying to solve issues caused by gene drive with another gene drive, right? And this is acknowledged in the literature as well. And a really big focus of my work is this issue of the risk of unintended spread, really an issue of containment, right? How can we ensure that early trials, just in case something goes wrong, aren't going to impact sort of, you know, broader global ecosystems? And this conversation about containment is really where islands suddenly become center stage, right? So you have organizations like the World Health Org and the National Academies of Science publishing these widely cited guidance frameworks for ethical considerations for gene drive. And a really taken for granted part of this conversation is let's use islands to contain this. So you see selections from these documents. Islands are described as ideal testing locations for genetically modified mosquitoes, particularly those with gene drive. Recommendations that the geographical isolation of the site can ensure there's negligible chance outside the impact, negligible chance of impact outside the trial area. And again, that islands constitute an ideal geographically isolated setting, physically limiting the dispersal of organisms. So as this comes on the heels of lots of conversation around this myth of isolation and sort of the harms that are continue to be associated with this framing of islands, I want to turn to Elizabeth DeLury here from her article, The Myth of the Isolate. She says, Pacific Islands have long been fashioned as laboratories for Western colonial interests, from the botanical collecting of James Cook's voyages to Darwin's theories of evolution to structural anthropology. So, as many have talked about today, we've seen these discourses of containment play out before in regards to islands, in many cases with devastating effects. In particular, also as many people addressed today, throughout the Cold War era, military powers, explicitly leveraged this language around isolation and remoteness to justify nuclear experimentation throughout Oceania. And as we know, the effects of these experiments were far from contained. So one thing I'm sort of paying attention to as I'm looking at these guidance frameworks and reports on ethical considerations for gene drive is that there's all this talk about stakes and stakeholders, right? But a sort of glaring omission within these discussions are, more critical framings of these stakes within the context of settler colonialism. And really there is, a big lack of attention paid to these parallels in language and framing of islands as isolated, and where we've seen that having devastating impacts before for islands and their indigenous peoples. So I'm gonna shift to talk about gene drive in Hawaii now, and continue this thread of these broader questions, more grounded in my ethnographic work. So as I mentioned, gene drive for conservation in Hawaii is being developed particularly at both campuses on Oahu and on the Big Island. And there's interest in engineering this mosquito, Culex quinquefasciatus, that transmits avian malaria to birds like Iiwi, who you see here, which is a Hawaiian honey creeper. And one concern that is brought up a lot is that these are high elevation birds. While they previously could, retreat to mid and high elevations to get away from mosquitoes because it was too cold for those mosquitoes up there, with climate change, increasing temperatures are expected to bring those mosquitoes up into those higher elevations and also increase the transmission of avian malaria at lower elevations. And so my research has involved ethnographic observation of community engagement events, or should I say community engagement events in air quotes. Also, I've done interviews with scientists working on gene drive and interviews with Kanaka Maoli on Oahu and Big Island to talk about gene drive. And so I'll just frame a few sort of preliminary findings for you here. One thing that comes up a lot in interviews I've conducted with Kanaka Maoli folks is that, when we're talking about experimental technologies, this frequently prompts comments and discussion about the many military and scientific appropriation of Hawaiian lands that have occurred and continue to occur since U.S. illegally overthrew the Hawaiian Kingdom in 1893. So folks bring up anything from decades-long bombing of Kaho'olawe, the biannual RIMPAC exercise in which military forces come from around the globe to test technologies on Hawaiian land, air, and water, and the University of Hawaii, 30-meter telescope, and also the University of Hawaii's controversial attempts to genetically modify kalo, which is an example I'll sit with for a little bit here, as I think it resonates a lot with thinking about gene drive. So in several interviews I've conducted with Kanaka Maoli, we've talked about, in 2003, University of Hawaii's decisions to genetically modify kalo or taro. And I have some excerpts from discussions here. We talk about, the sacredness of kalo to Kanaka Maoli, the Kumulipo, which tells of Haloa, the first Hawaiian. And I also draw on Noelani Goodyear Ka'opua here to remind us of something that comes up a lot in these conversations of why it's problematic to genetically modify something like kalo, right? And this is this reminder that the Āina is not something but someone, right? And I'm also drawing here on Miley Arvin's new book, Possessing Polynesians, in looking at this case. And she writes, while Kanaka Maoli argued that kalo was sacred and inextricable from Kanaka Maoli lives, and thus would be reprehensible to genetically modify it, researchers promoted that the genetic modification as a biotechnological improvement that was necessary to increase crop yields, improve pest disease, and advanced scientific research. And though a provisional 5-year moratorium on genetically modifying taro was passed in the state legislature in 2008, the Native Hawaiian case was often represented as simply a cultural one, whereas biotechnology proponents represented progress, delayed for now, but destined to win out, though through more culturally competent means if necessary. And so I'll just conclude with some thoughts to kind of tie up loose ends here, but what I'm thinking of in looking at these resonances across this case study with Kalo is that there's a really similar narrative I'm seeing playing out in gene drive research, particularly as scientists anticipate indigenous resistances to gene drive, right? We come across a lot of these framings of indigenous folks as merely uneducated on genetic modification, right? A lot of these engagement events are sort of organized around unidirectional education, right? We're going to do an education campaign and teach everyone about gene drive, and then they won't be skeptical about it. This is really mired in a deficit model, right? Saying the community, if they are skeptical, it's just because they don't understand, right? And within these narratives, also see a lot of sort of framings of indigenous folks as standing in the way of scientific progress, and this, resonates with a lot of media portrayals we're seeing on Mauna Kea as well. And so in harboring these views while also understanding the necessity of community engagement to try and advance gene drive research, something else interesting I've seen is sort of this co-optation of the language of participatory research, but also turning to like marketing as a mode of engagement, right? Like what's a palatable way we can present this technology, and folks actually even, entering into partnerships with research marketing firms to try and package up gene drive all pretty so that everybody will feel more comfortable with it, right? And so to conclude, I'll point out one more resonance that comes across in Miley's writing and resonates with the context of gene drive. And it's something that sort of gets read as a diversion when I present this work, for instance, to scientists, geneticists here on this campus and in conversations with scientists in Hawaii. And this is that scientists love to remind me, well, okay, kalo is sacred, but mosquitoes are not, right? Mosquitoes are invasive. And this line of reasoning sort of says, if gene drive can eliminate invasive mosquitoes and save the native birds, well, why wouldn't indigenous folks want that, right? And what I try to point out in these conversations is that these moves toward elimination, extinction are actually more reflective of a settler logic than indigenous ones. And as Ruben Gastambide Fernandez and Eve Tuck's writing contrasts indigenous and settler futurity, they explain that indigenous futurity does not require the removal of the settler in the ways that settler futurity requires the removal and erasure of indigenous peoples. And what indigenous futurity does seek, of course, is the undoing of settler colonialism and settler colonial epistemologies. And for me, this includes these logics and practices that I've touched upon a bit today that are very much at work within this field and this technology. So I'll leave it at that because I know I went over. Thank you, Sainamase, to the Speculative Teachers Collective.